Okay, so for the differences between Krebs cycle of respiration and Kelvin cycle of photosynthesis, right, the locations obviously are different. It happens in the matrix for Krebs cycle, but stroma for Kelvin cycle. Then for the overall reactions, right, we can compare, first of all, the starting material, that would be oxaloacetate versus RUBP. And then, right, what happened to the starting material? Okay, so oxaloacetate forms citrate with acetyl-CoA, while RUBP formed this unstable 6-carbon intermediate with carbon dioxide, which quite immediately, right, gets split into 2 molecules of GP or PGA. Okay. And we can also compare the regeneration of the starting material. That would be the regeneration of oxaloacetate after citrate goes through a series of oxidative decarboxylation, oxidation, and substrate level phosphorylation, while RUBP is regenerated from a series of reduction and phosphorylation. Okay, so the reduction, I think you are clear that is referring to the reduction of the PGA to TP. As for the phosphorylation, right, that happens during the reduction as well as the regeneration of RUBP from TP, right, because ATPs are required, right, so the phosphate from ATP is actually moved and added into the intermediate of the Kelvin cycle. That's why I call it phosphorylation. Okay. Right, so take note that for these overall reactions, right, we are not describing Krebs cycle and Kelvin cycle, right? We are trying to find features that are comparable, right, between these two cycles. That's why the things written here is a bit different from the usual outline that you have done before for Krebs cycle, right? So if you do an outline for Kelvin cycle, that also wouldn't look something like what you see here. Okay, so this is again comparison, right, between the two cycles, not describing the two cycles. Okay. Okay, then move on to the coenzymes for Krebs cycles. That would be the NAD and FAD, right? For Kelvin cycle, the counterpart that is parallel to NAD and FAD would be NADP, right? They serve different roles, right? For NAD and FAD in Krebs cycle, they remove or they take up the hydrogen, right, from the intermediates of the Krebs cycle. But for NADP, right, the reduced version of NADP actually provides the reducing power, right, for the reduction step in Kelvin cycle. Okay. For the carbon dioxide, that would be a byproduct in Krebs cycle, but for Kelvin cycle, there's actually a substrate that goes into carbon fixation. For oxygen, both cycles don't require oxygen directly, but for Krebs cycle, it only occurs when there is oxygen. So even though Krebs cycle does not require oxygen directly, it is only going to occur in the presence of oxygen because of the regeneration of NAD and FAD in oxidative phosphorylation in the presence of oxygen, right? But for Kelvin cycle, there's nothing to do with oxygen at all. For ATP, that's produced in Krebs cycle, but it is used in Kelvin cycle. And for the products, right, we have the ATP and reduced NAD and reduced FAD and carbon dioxide from Krebs cycle. Of course, you know the number of the different products per substrate. But for Kelvin cycle, we don't really know a number. It's a bit harder to pinpoint a number of products in the Kelvin cycle, right? So we will just name the different products without giving the numbers. Okay. So there's glucose, obviously, which forms from TP, right? 2 TP gives us a glucose phosphate. We have NADP after the reduced NADP is used in reduction. We have ADP after ATP is used in both reduction and regeneration of RUBP. Okay, so that would be just a quick contrast between the two cycles. All right.